Umapriya, can you show the slide of the next speaker? Yeah. So with this, we welcome on behalf of the Department of Biotechnology and organizing team. We would like to welcome our next speaker, Dr. Tanu Parmar, who is presently working in Case Western Reserve University, Ohio, USA. Dr. Tanu Parmar she had completed her PhD in National Institute for Research in Reproductive Health, Mumbai, India. And she did her first postdoc at Durham University, United Kingdom. And then she then completed 2 more postdoctoral programs in the same university, where she is presently working as a postdoc. So Dr. Tanu Parmar has 18 years of research experience, and she has published more than 15 papers in peer-reviewed journals, including JBC. The Journal of Biological Chemistry is one of the prestigious journal for any biologist. They know the value of publishing in that journal. So she has also publications in stem cell research, human reproduction, etc. She has received best presentation awards and travel awards from various agencies to her credit, and she has also presented at various scientific meetings held nationally as well as internationally. Her research area is on stem cells and reproductive biology. And as I already pointed out, amidst of her busy schedule, she has accepted our invitation to deliver the talk very immediately. I contacted her, and she readily accepted, and she also has prepared a fresh presentation, which is going to be explain you about the strategies. What are all the strategies that we follow from protocol to report writing? It's one of the important elements of any researcher. So we are going to have a nice session. So with this introduction, I welcome Dr. Tanu Parmar to give the presentation. Thank you.
And let me just, can you see my presentation right now?
Not yet.
Not yet.
Yes, it is loading.
Okay.
Yeah, you can make it as full screen.
Okay. Yeah.
Yes.
Is it working now?
Yeah.
Hi.
Yes.
First of all, thank you very much for this invitation, and I extend my huge congratulations to all the organizers of this workshop. As I was just listening to the last speaker, very comprehensive talk, sir. A lot of information. So, as this workshop has been basically organized to make everybody aware of 2 important aspects of science. Just doing science is not enough. Amazing science is very essential, but how science is documented, how science is presented is also a very essential criteria for success. So as the title suggests, I would like to share with you some thoughts on what I think of science documentation. So from protocol to report, what's in between. So just a schematic representation that these were very basic skills which we were introduced to in our very early years, that how to write like a scientist. You observe closely, you draw exactly what you see, you label the picture, and you write to teach about what you learned. So these are very, very basic steps which are still very instrumental, very strong. They still hold very strong as we progress further in our careers, that these basic skills we still need to work upon. So just imagine an everyday lab day. Oh, where did I put that piece of paper? When did I do that experiment? Where did I save that data file? When did I document the results of that experiment? So it's time I would call everybody to revisit these big questions that do keep popping up every day, whether you're working on the bench, you're in the field, or you're just doing theoretical science, that you just need to keep everyday track of everything you're doing experimentally. How you plan the experiment, what you do, how you present the data, where you document the data. These are small things, but they add up to really huge consequences. So a lab notebook comes into play. A lab notebook is a daily record of every experiment you're doing or you're planning to do or how you're going to execute the experiment. It's actually a daily record of your thought process of what are you planning about doing, what is the target of the experiment, how you plan to carry out that experiment. And then it becomes the basis of every paper, every publication and thesis or report you are going to be writing ultimately. So this is not just a personal record, it's a record which is very essential, which is used by patent offices in the courts of law when your lab is out to declare patents or to declare that you were the ones who first made the invention or you were the first ones to document this result. This is the very essential document, a lab notebook, which comes into play. So this is a record which not just enables you to reproduce what you did successfully, but also your successors or people who want to replicate or reproduce your experiments to be able to do it successfully. So this is a very, very essential piece of documentation in the form of a notebook. And this is also a very important scientific obligation which we have, because this allows your work to be reproduced faithfully, not just by yourself, but since science must be reproducible, this has to be reproduced by other people who try to follow your experiment. This facilitates accurate reporting and publication. It basically organizes how you do science, how you do good science, how you formulate your ideas, how you like to present your ideas, and what exactly are you trying to achieve by that experiment. So this helps in the basic planning of your entire process as a scientist. Why make a lab notebook? First of all, it is very easy to enter information stepwise. What you're doing, you just have to note it down, what exactly you were planning to do. If making lab book was really cumbersome, you wouldn't be keeping it up. It's very easy to find information when it has been put down in a sequential manner, and it includes any other information which you may want to refer later. So you always have something to fall back upon, not only for yourself, but basically to teach others or to help them reproduce the same thing what you did. So this makes it very essential to have a lab notebook. Not just us, but stalwarts in science before us have been having notebooks. Now, for example, I just put forth Leonardo da Vinci's notebook. Even after 500 years, we are able to look at those notes which he made. This is like the studies of reflections from concave mirrors, which one of the studies he did. He was a polymath, as we all know, not only an artist, but a scientist, a mathematician. We can still understand his notebooks from such long back and still deduce results from that. So this is really amazing. Another of his deductions, where in the second figure, he mentions about he did the proportions of the body. So this is one of the first initial human anatomical calculations which were done long back, thousands of years back. Even now, people deduce from that, people make conclusions from those things. So these are his notebooks, these are his record books in which he made in the form of figures, in the form of documents, and still people are referring back to that. Some of our known people, Darwin, Einstein, Pauling, these are just examples of the record books they made, and they put forth their data. They used to document each and every piece of calculation, every piece of information, every piece of experiment that they so neatly and so meticulously that these are still available to us. We can go through them and make so many conclusions which are still very relevant today. So I really found this very fascinating, that this is not something which we are asking the students to do now. Amazing people, like huge stalwarts in science, have been doing this and they've been doing it so meticulously. These are Alexander Graham Bell's notebooks, in which he had documented down the whole procedure of how he had made the invention. Francis Crick's notebook, where he had put These methods are still followed, and they were set down so clearly, so point-wise, and such neat protocols that they are still being followed. So coming back to the relevant, coming back to the current situation, how do you record the data? Directly into notebooks, that is one method, not on post-its, please, not on paper towels and scraps of paper, in blue, black, inedible ink, no gel pens. Make entries only in the ruled areas of the numbered pages. Unnumbered pages should not be used, only one experiment per page, and attach forms or printouts. So these are some of the methods, or some of the, everybody can develop their own particular personal method to record the data, but these are some of the basic backbones which can be followed when you're recording the data in a paper form of a notebook. Some of the rules, laboratory notebook rules, can be the entries should be made without skipping pages or leaving empty spaces at the bottom of the page. To start an entry of a new page, draw a line through an unused portion. Never tear or remove a page from a notebook. Each page ideally should be signed with the worker's full name and dated. All the photos, all the figures, computer printouts, and charts should be permanently put in your notebook with your initials and date over the tape. No entry should be changed or added to after signatures by a witness. And if the inventor or the scientist has any additional information, always a new entry should be made. Any mistakes there should be never whiteouts being used. You do not erase or write over your findings. You do not discard or replace, attach supplementary data. You always circle the reasons why you had edited the data, and you always add your dated initials to the corrected data after you have done the addition. Some of the things which can be kept in mind when you're making this very essential document, which we call the lab notebook. So some of the other rules which should be in place for a laboratory notebook are, you should always be having it is in a very safe location in the lab. Ideally, it's a property of the company or the university which you're a part of. And the old notebooks are also stored, and only specific destruction policies are followed for after a large number of years when these documents have to be destroyed. But ideally, they are kept for a very long time. So a good laboratory notebook is actually a record of both your physical activity and your mental activity. That what productivity are you are exhibiting in the lab as you enter your workspace. What are your plans of carrying out the data experimentations? So lab data, which you add in the notebooks, this can include gels. It can be scans of your HPLC peaks. It can be your photographs of the gels, their computer printouts, with all observations and calculations and conclusions written very, very clearly over your data. And all these data are very important to be preserved and organized in a very systematic manner. So what is a lab notebook? To be to go into more details, is that why experiments are initiated, what is the order in which they are carried out, what is your target on performing a particular experiment. There's a place to compile the data and the charts, to also troubleshoot the problem which you encounter as you progress through various steps of the protocol. And you also have a whole scenario to make out the whole picture of starting from why the experiment was carried out to the point where you get the results and the conclusion. So it's like a huge comprehensive process. So it basically becomes a to-do list for you. You have a recording of what you have done, but you have also put in the planning of what you had initially thought of doing. It clarifies the entire course of action of your experiment. It's a record of the protocols which you would plan to repeat, or you wish to have somebody else repeat it for you to have a reproducibility. It's a reference for others, as I said, people you might be collaborating with or your own lab mates when you want them to repeat the whole experiment. And it, of course, becomes a big support for your future publications, for your thesis, for your reports, for your lab, and for your grants, that this is how you want to present your data. So now with the more recent times, not just pen and paper, one can go digital. Electronic lab notebooks are becoming very key in meeting these research data managements. They are making things more easier. You can have files and records on your own laptop or your lab computers because the digital lab books, they have the ability, you can share methods a lot, you can share your data a lot, you can have backups, you can have copy and paste methods. So it becomes very easy for not only a collaborative effect with your co-partners or with your collaborators, but also a very good record for your own backups. So why electronic lab notebooks? As I said, this is now, we basically are using a lot of electronic lab notebooks. They are easy to read entries, definitely. They're easy to enter data. You don't have to type a lot. You just have to copy paste and do a direct transfers also. To search for entries for something what you have done in the backdate, it becomes much more easier. And of course, as I said, a backup is always like an added advantage of having an electronic lab notebooks. Multiple backups, I would say. So 2 levels of record keeping for scientists, basically, as I talked of, the lab notebook. The next step comes where you're actually putting out the contents of this notebook in the form of research reports. Now, research reports can be, it's just not like Word documents. You can have a research report in the form of grant. You can have a presentation of your research report. So the next output of your important documentation of a lab notebook is your research report. I'll talk about it a little. So the research report, the lab notebook being your working journal, the research report would be a specific organization which you are actually working towards a polished document. Yes? Hello? So these are the specific. Did somebody ask a question? I couldn't hear it. Hello? Hello? So,
yes, yes, Tanu.
Yeah. Did somebody ask a question? I felt I was interrupted by.
Oh, okay. No problem. You can please go on. We'll ask the. Ask the question. Answer
Okay,
the
sorry
question in
about
the
that.
end
I
of the
thought
session.
I missed somebody.
Okay.
Okay, I
Thank
thought
you.
I missed somebody.
Yeah, okay.
So as I said, the lab notebook is your working journal, and the next output is your research report, which is actually having specific sections in organizations, which is ultimate aim is to have a polished journal or a publication. So the various sections which I would say you should categorize your research reports in, there's a title, there's an introduction, there's material and method, results, discussion, and references. So this is like a general method which is used. So starting off with how the entire research is put out is I would like to point out some literature search engines like PubMed is the most common one. It's a free online archive of medical journals with about 25 million records in several databases. This is the most used to search engine for literature search in biomedical sciences. Ovid is also another comparable search engine. Now, this has, including PubMed, it has other databases like Medline and Embase. It has more than 35 million journals and which can be searched into. Web of Science is another popular one, which we use in biomedical research. This has a database of 8,700 international scientific journals from Thomson Reuters. Science Direct is another one with about more than 3,800 articles on science and technology and medicine. And Scopus is also one of a large databases which is used, which has about 60 million peer-reviewed literature, also owned by Elsevier. So see, these are some of the, as I mentioned, that now the step is on to have a polished journal, a polished publication report coming out. So starting off, is what you do is you do a literature survey, a literature search of the topic or the thing you're working on. So these are some of the popular search engines which you can use. So coming to from the lab notebook to the lab report, again, the title is the first thing. It's typically a single sentence that explains to the reader what the experiment is all about. You have the introduction in which you're giving all the relevant background information about your topic. What exactly are you following the narrative of your work? So it always lays out a logical argument as to what was the importance of the experiment, why it was being carried out, what was the lacuna in the field that you were trying to cover up with your experiment. Then the material and method, which is actually a list of detailed recipes or what reagents you used, what the methods, what were the animals or the human subjects which were being used. So this is actually detailed enough so that the experiment can be repeated anywhere in the world. So this should be so detailed. An essential aspect of scientific method is that the results are meaningless unless they're confirmed by others. So the material method should have all the details that it can be repeated out. And then you have, then you have the results, the statements of the fact regarding what the data was being collected with all the standard errors and standard mean and all the statistical methods being followed. And then you have the discussion, where there's an interpretation of the results and a discussion of its meaning. So in published research reports, the discussion will also put the work in context of other published works in the same field. And then you have the references, where you have like all the people or the literature search and the text you have quoted in your work. So there are a variety of formats which can be used. It all depends from journal to journal. So that particular format has to be strictly adhered to of the journal which you are planning to publish your work in. So some of the common mistakes which do happen, we still have to be very careful when we are writing reports, that we do not ever put the results in the material method section. Always there's a tendency to seep in some of the results while we are explaining the material method. This should not be done. You do not put any comments associated with the discussion section into the results section. This also becomes an overlap many times, but this is one of the common mistakes which people still conduct much down their careers. This should not be done, because someone might argue with your ideas in the discussion section, but there should be no argument about information in the results section. You do not duplicate said statements or paragraphs in different sections, because that becomes a repetitive thing. You do not ever duplicate data. If you're putting something in a table, you are not presenting again it as a figure also. And basically, the thumb of the rule, you must follow the formatting guidelines for the research report you're presenting, or you're putting in a grant, or you're giving a presentation of. There are some basic formatting guidelines and presentation guidelines which are given to the writer. They have to be followed, because any deviation from these specific guidelines, which are very hardcore, they will result in an immediate rejection of the report. So be very careful when you go over the instructions that what is required of your writer. Some of the writing tips which are like, I would say, I wanted to extend on my own, was that one has to be very clear and concise when you're giving out the content of the report. Do not go for flamboyant, buoyant language. There should not be any personal opinions. Stick to science. Data speaks for itself. You should not have any personal opinions about things like that. You have to support the statements with data from your experiment. You do not give opinionated statements without any evidence. We always write in past tense. Past tense should always be used while writing a lab report. And always it has to be reported in a third person, that it was carried out, we carried out that. So I would like to give forth some of the references in good writing practices, not just report writing, but also to maintain a lab notebook. Some of these references I would like to share on this presentation with the people who are listening to me. And some of this citing, I would like to cite some very good resources in scientific writing for which do stress upon all the points which I have been talking of. So this is what I had to share with all of you, and I would love to take any questions. Okay. Hi. Any questions? I would love to hear of any opinions if somebody has. Hello. Hi. Hi. Any questions?
Ma'am, exactly. I have a question. Can you hear me? I'm Dr.
Yes.
Achira.
No.
Yeah, I was really interested to see the lab notebook of all our scientists. It's really interesting.
Yeah, it was really very fascinating to see
Yeah,
that.
it's
Yeah.
fascinating. Whether they have the full paper copies of that in the, otherwise only certain pages they have, ma'am.
Yeah, so I was going through some of the web searches and I found these few clips. I'm sure that NLM or some libraries they do have full documentations of these notebooks of these people.
Okay. But exactly what you said regarding the lab notebook, most of the research scholars don't have this in the lab. And the key points which you have suggested is correct. They should have some, whenever they see some observation, they have to record it, or they are not
Yes,
doing it. That has to be done. And the way you took regarding the lab notebook, because your maximum of your talk was covering lab notebook, and it was really interesting. I was really interested in that one. Thank you so much. Thank you so
thank
much.
you, Dr. Shanmugan.
Dr. Parmar,
Hi,
this is Sankar Ganesh Arunakulam.
hello, sir.
I have a question.
Yes,
These days, we are using so much of electronic documentation. For
yes,
example, we take a gel picture and it is always stored in electronic form, right? In so and so computer and in particular folder, we keep it in.
yeah.
How do you think do you think this should be recorded in hard copy? This file has been stored in so and so, and what practice do you suggest?
I would say that please take a printout of that file which you have saved or the gel picture or whatever and paste it somewhere in hard copy, because nowadays every journal, every big journal is actually demanding to see the actual gel picture or actual hard copy of that. They are not just satisfied with scanned images. This is something which, at least in US, we are encountering this, that big journals like JBC or Nature or Science, they want the real picture there. They want to see it. They actually ask us to post those pictures to them. They're just not happy with the scanned images. If there's a Western blot, if there's a gel image, they want to see the whole. So even a copy of so I would say take a printout of that, scan and paste it somewhere where if you have to show it to somebody specifically, the public, for the publication purpose, you should have a copy of it.
Yeah, exactly. We always post it on the notebook, but in
Yeah.
addition, do you think is it important to note it down there as like F folder, F for drive, and this folder
Yeah,
and this
you
file
can have
name?
your, yes, definitely, because as I said that now the trend is to have electronic notebooks with a copy of your own. So it has to be labeled and titled very properly for your own records, and parallel to that, pasting it in your hard copy, that in your notebook, actual paper form notebook also. So yeah, this is like extra work to be done, but yeah, it is essential. Specifically, if you're really planning to have high quality journals publishing, then questions can be asked at any point. They might ask to see your raw data. So this double work has to be done, I would say.
Yeah, thank you, Dr. Panda.
Thanks.
Hello.
Hi.
Hello. Hello.
Hello.
Yes, sir. You can ask the question. Yes.
Hello.
Yes, sir. We can hear you. Please ask your question.
Madam, excuse me. Hello.
Yes. Yes.
Is it
Yes,
audible?
sir. I can.
Yes, madam. Very nice presentation. I have one doubt. Most of the time, what we are getting results. Most of the time, we are getting negative results. Such kind of results usually not published in journals or like that. So if it is the condition, what we do, sometimes whatever the results we are expecting, we can't able to get, then it become negative results. Even most of the time, even researchers, they are not allowing to present the negative results.
Right.
What is your opinion about whatever the negative, about that negative results?
So no result has to be ignored. Ideally, if you know of it, there's even a journal of negative results. I'm not saying that you got to publish in that, but see, even the negative results are going to help you understand the process of what went wrong in your experiment, right? You have to refer to that also. So even the negative results, I would like specifically appeal this to students that please document everything, even if it's a negative result and you're not planning on presenting it, or even not leave alone publishing it. You're not even telling about to your PI about it that okay, we didn't get the right result, but everything has to be documented. So nothing should be kept undercover, because a real scientific process requires everything to be out in the open, because you don't know what clue you might get from that negative result also. You go back to your results after 1 year and you find that, okay, this went wrong somewhere. Or maybe this was pointing out to an hypothesis or a mechanism which we never even thought about. So I would say even a negative result might lead to some very interesting observation which we might have ignored at that time. So please document that also. That's what
Thank,
I believe in.
okay, thank you.
Thanks.
Dr. Parmar, there is one more question from Rajesh Kannan. What are all the common errors in writing discussion?
So as I mentioned, often the results and the discussion section gets overlapped a little. People tend to intermix or repeat things all over. Discussion should, I would say, highlight what future you're giving to your science, to your logic, what you were planning to do, or what lacuna you were planning to fill up in the field by your experiment. The uniqueness needs to be pointed out in discussion, that this is what was not known and this is what we are presenting. So I would say discussions should stress on that. That what is the novelty of your work?
Yep. Thanks for answering it. And any more questions from participants?
Hello, sir. This is Vishwanathan.
Yes, Vishwanath,
Hi.
please.
So my question is, we are publishing a research protocol, right? Hello?
Yes, yes.
Hello?
Hello.
Yes,
Yeah,
we
I
are
can hear
hearing
you.
Vishwanath.
Yeah.
Please go on.
Whether we publishing research, yes, sir. Whether we are publishing a research protocol, will this the protocol is challenge to researcher in publishing their original finding after the completion of their studies.
Sorry, I didn't quite catch the question.
Whether publishing the research protocol will challenge the researcher in publishing their original finding after the completion of their studies or their certain part of the studies in their thesis.
Sorry, I didn't
Okay,
hear the
so
last.
he's asking, yeah, I can clarify the question. He's asking something
Okay.
like we can publish the protocols, right? In some
Uh-huh.
journals.
Yes.
Yeah. So
Yes.
he's asking something like
Yes, sir.
after publication
Yes,
of the protocols, is it possible to have a good publication with having that protocol inside in a manuscript, whether that manuscript can be published or not?
Manuscript, yeah.
So,
Yes, sir.
personally, I have not come across thing like that, so I would not be the right person to comment on this.
Okay.
Sorry, I don't know.
Okay, no problem.
Hello, ma'am.
And one more question: how do we discuss the results which are not what we expected or not in par with the standard?
So you are the person who knows your study to the maximum. So basically, you know that what you are filling up in the field, what is it novel that you're bringing to the field. So when you say that your results were not at par, are you trying to compare it with studies which are out already published, or what do you exactly mean by that when you say results are not at par?
Yeah, he or she can post the question in another way with more
Okay.
clarity. And one more question, please tell us something about the summary and conclusion. I think she's asking for the difference between a summary and conclusion.
Between a summary and okay, summarization of your at the end of your report would be basically having a little bit of all your, not your title, your introduction, basically an abstract, which I didn't elaborate a lot in that. So it basically starts off with with a few lines of your introduction of the methodology you followed and the result. But conclusion is basically only the results part. So summarization would also have a little bit of a discussion of your result, what you finally got in your got at the end of your experiment, and conclusion would not be having that.
Okay. So the next question is, while writing summary part, is there any necessity to have all the results?
I would say only the major or the salient results can be highlighted. You don't have to go more descriptive. You can just outline them, like one or two sentences.
Okay. And one more question from Sasireka Manikandan. So she's asking whether results photo is getting importance or the tabulation form. Which one is best?
So actually, every journal has very specific guidelines of what they want, how they want the data to be presented in. Some want more tabular forms and some want more figures. Sometimes color figures, or they are more expensive. People try to do it in tabular form. To try to see how strong your data, the strongest form where your data can be presented. You know whether a figure will be highlighting the result maximum, or a tabular form will be doing that. So you have to be first judge of that, and then you have to see the journal's guidelines of what they want. So both things have to be kept in mind about that.
Okay, so that question was refined by one person. So he's asking like, how do we discuss the results which are not expected? So it's something like when we do some research, it is completely different from the standard what we are using. So in that case, how to discuss those kind of results?
So when you say they're different from the standard, what do you mean by that?
Yeah, for example, if you are treating a particular disease with an animal model, right? We are using a standard drug and we are formulating our own drug and doing experiment in the animal. So the standard drug is giving some good effect. Of course, it is a standard drug, but when your formulation is not giving results close to that standard, or if it is much deviated from your standard, then how do we discuss those kind of results?
So if you can give a logical explanation of why, how, if your deviation is actually statistically significant, you can always find a logical explanation of how that deviation got you to that result. So I think the presentation would be exactly quite similar as you would do normally in a result which was not away from the standard or different from the standard. So I think basically go for first that was it statistically significant or not. And if it is, then I think you go by the same route that you explain that why the result got deviated. Science always has a logical explanation of why things had happened in that particular manner.
Okay. The next question is, what makes a good research statement?
A good research statement. So it would vary in different scenarios. Like for example, if you're making a research statement for presenting a grant, you're writing a short proposal or a short grant, or you are planning to present it out as a presentation, or you're submitting for, like, say, an award grant. So a research statement basically should, I would say, 2 points. Firstly, it should be able to add something to the field. I would say a novel aspect to the field, or highlight something which was not existing in the field, which is what you are bringing out, what your study is bringing out, basically that, and what future conclusions can be made from that. So I think anything which can bring out the novelty of your work, which was not there in literature, which had not been worked upon, is something would make a strong research statement.
Okay. So the next question is summary and abstract same or different?
Sometimes I use interchangeably because, as I said, that unlike an abstract, the summary has a little bit of everything. It has a little bit of introduction, the methodology we used, and the conclusion which had been drawn from that. So according to the format, as I said, that some journal articles or some research presentations, they require an abstract. Sometimes they don't need a summary, they just go to abstract and then they want conclusions. So this can be used interchangeably. So just go through what the guidelines of the requirement is, that what the journal or what the proposal is demanding of you or what they require from you, and give accordingly. But basic essence is that they are quite interchangeably used. They're quite similar.
And one more question from Kishore. So he's asking, abstract can be in a flowchart or in the form of picture representation while submitting for the journal. He's asking something like graphical abstract and written abstract.
So the same thing. First, the format of the guide of the journal has to be gone through. Sometimes they do not want any graphical presentations. Like you're making a PowerPoint presentation or you're giving a lecture, which is like in the form of presentation. There you can be more experimental. You can give it in the form of a graph, which is, or a tabular form, which is like more understandable, to a larger crowd. But when you're submitting for a journal, you have to very strictly adhere to what the journal demands of you. Mostly they are in the form of a word form. They do not need graphical or tabular forms of the abstracts at all. At least I have not come across any of them who have like asked for like or allowed even a graphical presentation. But yeah, when you're presenting at a local international meets, or you're presenting a poster or giving a talk, then you can be more experimental with that. Give it in any format, like a graphical form or a figurative form, which presents out the whole summary or the whole abstract more clearly. But while submitting in a publication, you have to stick to their guidelines.
Okay, so this can be considered as the last question, I think. So this question is from Rajesh Kandan. Whether detailed methodology is essential for publication?
Yes, it is very, as I said, that material and methods section would enable the basic reproducibility of your work. If anybody has anywhere in the world has to reproduce your work, he or she would need the exact details directly to, like, down to the molarity of the solution which you had used, if you can put it in the supplementary data behind. So yes, material and method, I would say every journal appreciates the more extensive, the more detailed it is, it's better. So even if they're not asking for it, give even more. So they have to be really very, very detailed.
And one more question from Shiva Sakti. She appreciated your presentation and she's asking, how will be a graphical abstract help in explaining a study or a review, or how can we do it do a graphical abstract in an effective way?
So, as I said, disregard sending in publications, but when you're presenting your data, a graphical abstract can be, I would say, it would be much more informative, because you can start off with your arrow point. For example, you can start off with your arrow pointers, or with your some shapes and figurines can be started off with. You start off with, this is the aim. This was like the target of the experiment. Then an arrow shoots out down. This was like in a circle form, material and method used by it. So you can use a lot of shapes and like arrow pointers to make it in the form of a graphical or a figurative form. So I think as you are wishing to do, a figurative or a graphical abstract would be much more informative than just writing out words. So you can be very creative in that. With the use of arrowheads and shapes, you can make it very concise as well as more informative to deal with.
Yes, thanks. Is there any more question from the participants? Okay, if there are no more questions, we will share the email ID of the speaker, so you can get in touch with her if you would like to have any more questions related to these aspects. And at the outset, I would like to thank Dr. Tanu Parmar for your acceptance for giving the talk. And it's almost 2:30 in the midnight, right?
Yeah,
Yeah,
fine.
it's very hard. It's very hard because most of the people whoever I contacted in the USA say they simply rejected due to the time difficulty. So I missed off your busy schedule and busy time, and you spend a lot on the preparation, as well as you did a great job in doing this for the research community. And thank you very much for your presence
Thank you,
over
Dr.
here.
Dinesh. It was a
Yeah.
pleasure to be with all of you, and all the best to all the young people who are in the crowd.